Hello, welcome. We are live for the first time on YouTube. Super excited to be studying with you to get ready for the AP Bio exam. It is so soon. So soon. I mean, obviously, AP exam started across a lot of classes this week, and I know that we're all a little stressed. So if you're taking the time to come study with me tonight, I really appreciate it. A lot of good information that's going to be coming your way. But let's start with introductions. So I am Melanie Kingett. I'm from the Absolute Recap, and I am the host of a handful of things. So here we have, just to get started, we're going to be doing the binge, right? And so since the Absolute Recap started with a podcast, we are binging the content, right? And so whether you are here from the podcast or whether you're here because of the ultimate review packet, we're all getting ready to prepare for the exam. So first and foremost, welcome. There's a couple of things I want to make sure that you have at your fingertips in order to be ready. The study guide. Okay, so if you look at the top of the comment box, you can see the link that's included to make sure you can download the study guide that goes with this presentation. In just a second, I'm going to walk you through how to get it, which take only a couple minutes to download. And once downloaded, you'll be able to interact with the presentation, take some notes on the side, and really get ready for the test. Second, let's use the chat. I want to make sure that we're being respectful, right? Everyone's here maybe with a different level of content knowledge, and that content knowledge can be a little unsettling, right? If you hear someone else that might seem like they have their answers really snappy and they have the information at their fingertips, and maybe that's not you yet, that's okay. Let's help each other. So please be respectful in the chat. I will answer as many questions as I can as I see them come through. Our structure tonight is going to include some practice questions as well. So if you think you know the answers, pop them in the chat, and we'll try to work out some of those misconceptions. So let's talk about how to get this study guide. Once you click on the screen at the top of the chat, you're going to see a link to take you to the ultimate review packet. And so this ultimate review packet will add you directly to the ultimate review packet page to biology. When you are on the page, you can just take a quick scroll and see that the URP includes a lot of things, including all 8 units. We have our practice exam and answer keys. But most importantly, right, what's included with a free preview? That third tab down says live exam review. And whenever you see that free preview button, that's going to tell you that you don't need to have any access to the URP other than signing up for a free preview account to get these resources. So the study guide that you want to download is right here, units 1 through 4 live, the review study guide. And in order to get that, you're going to click free preview at the top, and you can log in with either your Google account or make an account. But you can see that the price here is $0. When you have that downloaded, you will be able to access the study guide, right? The study guide is going to include information on these slides, some of the practice questions, as well as a place for you to take your notes. So with that, let's get started. Once again, I'm Melanie Kingett. I'm from the Absolute Recap, and we are going to be able to review today. The podcast resources, you can really access them anywhere. We're across YouTube with the podcast. We are also going to be over on the Spotify, Apple Music, Amazon has a playlist streaming service as well. All of that is going to be where you can download the podcast. We have over 86 episodes. Obviously, if you're here on YouTube, we are going to be using YouTube as well. And there's a lot of content on here with more coming in the future that you can access at any time. And of course, the ultimate review packet, 3 of the resources that we have to help you get ready for the exam. But I need to take a quick second and shout out my friends. There was a really funny comment the other day online about Mrs. Tiffany Jones at AP Bio Penguins, and that in some way perhaps we are rivals. We are actually very much social media friends and YouTube friends. Tiffany at AP Bio Penguins. I'm sure there's some AP Bio Penguins here tonight joining us for this live. So if you are looking for some free resources, if you're looking for just like a really good emotional support for the exam, I've linked her website down in the description. You can access that. She has tons of quizzes, really responsive on Instagram, has a Q&A box that she will answer your questions for you. So check out that for sure. The other thing that I want to make sure you are accessing is deep learning. So deep learning is an AI tool for a variety of AP subjects. They have teamed up with content creators like myself that will allow you to ask content-specific questions, and it pulls information from all of my scripts, right? So anything that I've written for the podcast or for YouTube can be used with deep learning. And that also has a quiz function, some FRQ grading functions. And so I've also linked their resources for you down in the description below. So feel free to check those out too. And my other friends at the URP, right, the bottom of my YouTube page, it'll have all of their content pages linked, everything that they have free for you. So if you're taking AP Bio, you're probably taking another AP exam. And that's going to be an important thing for you to really access as much support as possible. And many of the resources are also free. You don't have to necessarily get the ultimate review packet, but if you're looking for extra practice, you can absolutely do so. All right, here we go. Let's zoom in. One of the biggest phrases we use on the podcast, getting down to those details. So we're going to cover units 1 through 4 tonight, and I specifically have the percentage breakdown that the CED, that's the course exam description, has for each of those units. The course exam description is like the holy grail of all of the AP classes. It's not a secret. It's not just for teachers. You can Google it. It has all of the core content and the core skills that you need to know for each AP exam. That being said, there is more that you have to know that's not in the CED. It's just kind of like the baseline information for you to apply. Based on all of these breakdowns, you can see chemistry of life really gets the short end of the stick, but the content from chemistry of life unit, it shows up throughout many of the other units. So although it doesn't have a lot of emphasis individually, and really it's never had, as far as I can recall, its own FRQ, it's going to be integrated into other content. We're going to go through unit 2, 3, as well as 4. And typically unit 3 is not everyone's favorite. We'll kind of take out some of the scary from cellular respiration and photosynthesis for you in our conversation tonight. All right. Unit 1, chemistry of life, zooming all the way in microscopically on a molecular level. So learning objectives, these are directly from the CED. I'm not going to spend any time on them at this moment, but you can see that these are the goals of the unit. I have them copied for you in the study guide, and so you're welcome to see those as you download that document. But I am going to pause here because as a teacher who constantly gets asked, Mrs. King, do we have to know this for the test? There's always the, what don't I have to study? Can I take any shortcuts? Can I just not worry about some information? And the answer is yes. So the AP CED actually excludes information and tells you things that you don't have to know. So in unit 1, this is going to be specific structures of nucleotides, right? So you don't have to visually be able to tell the difference between adenine and thymine within a nucleotide. You should know they're one-ringed, two-ringed components, but structurally, where are the nitrogens? That's not necessary. Amino acids. You don't have to be able to tell me what methionine looks like. But if they show you methionine on a diagram and tell you it's methionine, then you should recognize, oh, here's the R group. Here I have my carboxyl. Here's my amino group. All of those pieces of amino acid structure are important, but not the specific amino acid itself. The same is true for polysaccharides, starch versus cellulose. They'll give you the diagram. It will be labeled, but you shouldn't have to identify what it is individually. And also the same is true for lipids. So if you've had to do that in the past for your teacher, just know it's not an expectation for the exam. All right, can't start without water. Now, water is one of the most amazing molecules on Earth, and it forms polar covalent bonds within the molecule of water. And so if we look at the labeled diagram here, which I drew, we can see that there are extra valence electrons on that oxygen, which is not being bonded to the hydrogen. And so the oxygen is more electronegative. It's actually going to hold those electrons closer to the oxygen atom as compared to the hydrogens. And so we end up with a molecule, although it's not charged, it has an uneven distribution of charge. And that's going to mean that we are going to create a molecule which has opposite sides, a molecule that has one side that will act one way and another side that acts another way. This is the foundation of hydrogen bonding, because hydrogen bonding is a weak intermolecular force. It's not actually a bond, but just like the starfish is not a fish, hydrogen bond, not a bond, but it will cause water to have really distinct properties. Those properties are cohesion, right? When water is attracted to other water molecules. We have adhesion, water attracted to other things that either are polar or charged. And this creates lots of properties like surface tension, high specific heat. It also causes ice to have a lesser density than liquid water because we form a crystalline structure when those hydrogen bonds push apart. But water, it's amazing. It's going to be involved in lots of our chemical reactions, including hydrolysis and dehydration synthesis. And so thank you, water. All right, moving into our macromolecules, we have carbs. Now, 1 to 2 to 1, that is our carbohydrate elemental ratio. So if we think of the classic glucose, C6, H12, O6, this is going to be important to the either ringed or branched structure, but carbs are only going to have those elements. Now, when carbs join up, like for example, here we have sucrose, you're not going to have exactly a 1 to 2 to 1 ratio, but that's because we have had dehydration synthesis at that glycosidic bond. We are in monosaccharides as our monomers, forming polysaccharides as our polymers. They are polar. And they have a lot of functions. So glucose, I already mentioned, is going to come back again very shortly. And you can also have structural carbohydrates like cellulose in plant cell walls. Proteins, big fan of proteins. When in doubt, it's a protein. That's what it does. And so when I look at my monomer structure here, I find an amino group. I have my carboxyl group. There's a hydrogen, and then the variable R group. And that's where the plug and play will matter. Very few amino acids actually have sulfur. But that's why I kind of have it in parentheses. That's not super common. Nitrogen in our amino group, one of the 2 places we see nitrogen in our macromolecules. But C, H, and O, they're in everything. So they're not really going to give any clues as to which molecule you are looking at. The, excuse me, the parts that we see on the side. So the way in which the protein is going to fold is really significant because this is going to influence the behaviors that our protein has. We want to be very cautious in the way that we describe proteins losing function. Structure influences function, huge theme of biology, but the structure needs to have very specific pieces, like literal locations in which smaller units will bind. When you take a protein and you have a very specific fold, it's going to fold that way because of the R group interactions and those hydrogen interactions that we see with my two sides of my amino acids. The amino group and the carboxyl group, those are going to be bonded to an adjacent amino acid, forming a polypeptide chain. So those aren't going to influence folding. It's those other elemental pieces that will. When in doubt, a protein does it. We're going to go through protein a ton when we do protein synthesis. I see someone asked in the chat, are we going to have a unit 5 through 8? Absolutely. It's next week. I think it's on Tuesday. I will recap kind of all of those pieces for you later on. Just backing up in the chat real second, I was having a technical glitch. So I want to say hello to everyone. This live is going to be an hour. I'm already going too slowly, so maybe an hour and 20. It will be saved. AP Bio Penguins recommended me to you. Oh, I love that, Kyle. Thank you. She's the best. San Diego, hey. Awesome, awesome, awesome. All right, we are going to do a couple practice questions coming up super shortly, so keep going in the chat because that's exactly where we're going to want to put our responses. All right, lipids. Lipids are nonpolar, except for phospholipids. So lipids are CHNO. You're going to find a greater ratio of hydrogen to oxygen than you saw in carbohydrates. It's one of the clues that you have a lipid because like the straight chains can look really, really similar. When we look at lipids, there really is no true monomer because there's just a lot of diversity, right? You have fused carbon rings. When you have steroids, you can find here triglycerides and also phospholipids, right? Phospholipids kind of steal the show, but it's the phosphate group, because it has a negative charge, that makes this molecule amphipathic, meaning that the nonpolar tails are not charged, but we're going to find a polar head due to that phosphate group, which will cause the bilayer to structure as it does. And I just want to point out here in this diagram, we have two types of fatty acid chains, one with a double bond, which is going to be unsaturated, and one that is straight, right? So saturation refers to a literal elemental saturation of those hydrogens. Function here. Energy storage structure, ligands, which we're going to mention very, very shortly. If you think you remember what a ligand is, let us know in the chat. And we will discuss that when we get to communication. All right, last one, nucleic acids, CHONP. They have every element of CHNOPS but sulfur. And here we are concerned mostly with directionality, number one, as well as bonding patterns with our nitrogenous bases. So directionality, I'm specifically referring to the order of the carbons on my carbon ring. That is going to influence whether I am 5' to 3' or 3' to 5'. So the phosphate group is going to be attached to the 5' carbon, whereas the adjacent nucleotide is going to be attached to the 3' carbon. Coming off of my 1' is going to be the nitrogenous base, either purine or pyrimidine, single or double ringed. Difference between RNA is going to be uracil. We also have double versus single stranded. We're going to regroup on nucleic acids again later on, so I don't want to give them too much attention right now. Signaling molecule. Great job, Sophia. Yes, a ligand is a signaling molecule. All right, let's get ready to practice. So if you have the study guide, this is a great place to interact with the diagram. If you don't have the study guide, still not too late to go grab it at any point throughout this presentation. It's linked at the top. It's totally free. It's a PDF download. It's 8 pages so that you can back up with all of this. Don't worry, I see a comment here. I don't remember a thing. This is Unit 1 stuff. I personally do Unit 1 in like 8 days with my students in August. So if this is a little bit fuzzy for you, it's also a really small portion of the exam. Looking at a 2020 CED question here, scientists examined the folded structure of a purified protein resuspended in water and found that amino acids with Non-polar R groups were primarily buried in the middle of the protein, whereas amino acids with polar R groups were on the surface of the protein. So of that, which of the following best explains the location of amino acids in the folded protein? Information about non-polar R groups kind of huddling up in the middle. And we have polar R groups on the surface of the protein, so kind of sticking outward. Couple options here. If you think you know the answer, please pop it into the chat. So 2 choices justifying the polar R groups being on the surface because they can form ionic bonds with charged things. Polar R groups are too bulky to fit in the middle of the protein and are pushed towards the protein's surface. Non-polar R groups cannot form hydrogen bonds with water or push into the middle of the protein. So here we have some repelling action. And then non-polar R groups from different parts of the protein form covalent bonds with each other to maintain the protein structure. So why is the protein folding in this way? A couple votes for A, I see a couple votes for C. All right. And C is the best answer of this question. Yes, they cannot form hydrogen bonds. And so part of that is going to cause a being repelled from the water molecules and pushed into the middle of the protein. This is a really similar reaction to what we see with the phospholipid tails, right? And as to why they kind of huddle in the middle of that bilayer. Great job, everybody. Okay, Unit 2. I was fast, I know. There are no elephant-sized amoebas. Cells have to stay super, super, super small for a lot of reasons. The amoeba is actually one of the mascots of the Absolute Recap, so we are fond of amoebas here. And although they are one of the bigger protists, they are not going to ever get so large that they can't function, right? That would be not ideal. All right, learning objectives. Let me get a little larger here so you can see them. Double digits. You can see Unit 2 has a ton of information. This is all on your study guide. Here's what you don't have to know. You don't have to know specific smoothie R functions, like how it's involved in lipid synthesis, how the Golgi is going to be involved in that as well, or how it packages specific enzymes for vesicles. It's just a level of detail they're not going to ask of you. But I will be honest, many students who take AP Bio have taken, look, I'll call it baby bio or Bio 101 before the course. They learn some bad habits when it comes down to the organelles. So don't mind me while I soapbox briefly. As an AP grader, I see a lot of correct information that will not earn points when it comes to organelles. And so this is often due to students simply restating a function of an organelle or some sort of analogy of what the organelle does. Unfortunately, on the AP Bio, it's not going to be deep enough for this. The ones that have asterisks come back in later units. So in terms of like what the organelle importance is, they repeat multiple times. And so it is going to be more likely there'll be questions on those since they show up in multiple units. My advice to you, clearly you should know what the organelles do. But think about it from a different perspective, right? So imagine the organelle breaks, right? There's something that goes wrong. What's going to happen as a consequence of that change, right? So if something is interfered with or there's a mutation of instruction, how would that impact the relaying functionality of that organelle? And so when we look at some example questions, that's what we will see. Question I saw over here, do we have to know how to draw the macromolecules? You do not. That is not an expectation of the CED that you have. Kyle, don't be afraid of amoebas. It's okay. All right, let's get right into practice. Let's see what we remember about our organelles. Here we have a pathogenic bacteria that's been engulfed by a phagocytic cell as part of a non-specific innate immune response. Which of the following illustrations best represents the response. So if I were to look at all these diagrams right off the bat, a few things I'll notice. This poor bacterium is being engulfed, some sort of pseudopods being wrapped around it. It's being brought in by a vesicle. It's also leaving every choice in the same way. So nothing about its entry or exit is different. So here we're left with ribosomes in A, antibodies in B, C has lysosomes involved, D has the mitochondria. So of these, which of these organelles is most likely going to show how this immune response is going to function, right? So you don't have to know the immune system for the exam. You actually don't have to know any body systems for the exam, but this is a good example of applying your information to the concepts you do have to know. A lot of votes for C. Let's take a peek. The answer is C. Ribosomes are involved in protein synthesis. Antibodies are not a non-specific response. So those are structural proteins that will be designed to specifically attack, and you typically break down an invading foreign body. And the last choice here is the mitochondria. Mitochondria involved in cellular respiration, so that's not going to be involved either. So the lysosomes are like Lysol. Angela, thank you for that analogy here. Great way to remember that they are involved in intracellular digestion and some sort of cleanup function. Just make sure that you don't use the analogy on the actual exam, but whatever helps us to kind of trigger our brains, I'm all here for it. All right, surface area to volume ratio. And this is why there are no elephant-sized amoebas. Surface area is a 2D concept, volume is a 3D concept. And so as we get larger, there's going to be a limited ability for the cell to be efficient, right? So if you get too big, you can't take in food and nutrients fast enough to kind of nourish the core of your cell. If you get too big, you can't get rid of waste fast enough. And by fast enough, I mean either through diffusion, facilitate diffusion, active transport, whatever your mechanism happens to be, still has to go through your barrier, right? Your plasma membrane. And so I like to think about this from a perspective, like if you're going to some type of sports arena or game, and you're exiting the stadium and they only have like 3 exits open, you're in traffic for at least an hour. The concept of that is that if you have to wait in line to go through a specific barrier and you're actually like a toxin, well, then the cell is not going to survive. So surface area is that literal physical space that has to be moved through in order for the cell to achieve its metabolic functions. This is on the formula sheet, and I'll be honest, it's there. It's rarely used by students during the exam. It's kind of like a formula, like security blanket, if you will. Don't memorize any formulas. It's just not, well, really don't memorize most of that, but it's really just not, it's not a good use of your time. It's you can reference it at any point throughout the exam. But surface area to volume ratio is really why so many things have folds, indentations to allow for greater activity across those surfaces. So the mitochondria is a great example. The inner cristae, that inner membrane, highly, highly, highly folded, meaning it has a lot of membrane within the organelle. That's going to allow for a lot of ions to move across that membrane, a lot of proteins to be embedded in that membrane. And that's going to allow the mitochondria to function the best that it can. All right, let's move on to our next Unit 2 concept, an unlabeled diagram. What was I thinking? Okay, obviously, the phospholipids are there with blue and black tails. Does anyone know what the yellow is in this diagram and why it is placed where it is? It's not on the perimeter of the, it's not on the perimeter of the phospholipid membrane at all. It's inside the tails. What is the yellow in this diagram? Pop into the chat if you know. I'm going to focus on the purple while you guys answer that question. So here I have two purple structures, one being hot dog shaped, looking like a protein channel formed from one side of the membrane to the other. The other is a peripheral protein being involved in some sort of attachment or cell signaling. Great job. Yes, the yellow is a steroid. It could be cholesterol within the membrane, influencing fluidity of that membrane. All right, and lastly, we have the green in the diagram. These are going to represent carbohydrate chains involved in cellular recognition and at times cellular signaling. So you'll find those as well in the cells. Cell diagrams can vary so dramatically. I like to draw my own because I do find that it allows it to be present in a way that I don't get stuck on one specific version of what it should look like. And the likelihood is you're going to see things on the exam you've never seen before or are represented in a way that you're not as comfortable with. So exposing yourself to lots of different diagrams, and at times, honestly, they're like excessively simplified on the AP exam because the fluff, like the extra stuff is gone. They just want you to focus on specific things, and they might not even label, like structurally have them. It might just be a label, like this is what this is in this space. So I do encourage you to look at a couple of different diagrams for different things. Try to caption them in your own words as we go through. All right. Yes, those carbs are for cell identification. They can be involved in that kind of like cell labeling as well. All right, looks like my slide got a little wonky from the transfer. It's not quite lining up here, so I might have to verbally go through a couple of these things. I apologize for that. It looks like it didn't transfer well. So transport through the membrane. We have two categories here, that which uses ATP and that which does not. So no ATP used means that the reason molecules are going to cross the membrane is that they're going to go from a high concentration area to a low concentration area. This is passive transport. It can occur with these molecules moving directly through the membrane, like osmosis or diffusion, or through a protein, which would be facilitated diffusion. Difference between the two is if you're going to pass directly through the membrane, you must be small and nonpolar. Water, although polar, super small. And in small amounts, it can fit in between phospholipids. If you need to move a lot of water all at once from high to low, you're going to have to move through a protein. It's called an aquaporin. So that would be classified as facilitated diffusion, but it's still all passive. And the reason that these molecules are moving high to low is really just they're bumping into each other. There's a lot of kinetic energy as the molecules go from an area where they're more crowded to where they are less crowded, and they are more likely to go from high to low. It doesn't mean they will exclusively go from high to low, but they're just more likely to move in that direction. On the other side, if you have to use ATP, you are going to be pushing molecules into an area where they are already more crowded. You're going against their concentration gradient. Doesn't necessarily mean they're even going to be able to fit over there. They're often going to be large or they're charged. Moving through a protein, I think the most famous, I guess, if you could be a famous protein pump, sodium potassium pump gets a lot of glory. Moving sodium and potassium ions, which are charged in opposite directions across the membrane. And then we have other movement through vesicles, endo and exocytosis. That example that we just had with the bacteria being engulfed, that was going to be an example of endocytosis. This diagram is not wonky in the study guide, so apologize for that, but you can use a study guide for the correct one. All right. Osmolarity is a scary concept for some because of the math. Water potential calculations are on the formula sheet. And I know I'm not going through those calculations tonight. We're going to do a little bit of that in the final review if you're tuning into that the night before the exam, just kind of some last minute math. But I have some broad notes here. First of all, tonicity, when we're describing hypertonic, isotonic, hypotonic, we're specifically describing solute concentration. And so the concepts that we use with that is going to influence two things. If the solute can move, right? If it can move across the membrane, in which direction and why, right? So high to low is going to be the general movement of solutes from hypertonic to hypotonic, if the solute can move. Water is going to move in the opposite direction because water follows its own concentration gradient. So if you have a hypertonic solution, it means you have a lot of solutes, you must have a lesser concentration of water. You have a lower water potential, less free water available. And so water tends to move in the opposite direction as my solutes. But what's important to note here is the water is going to go whether there's space for it or not. And so these are going to lead to situations of lysis at times. If water moves in faster, then an organism can maintain its balance. So we see this at times with paramecium. Paramecium are normally in saltwater environments. So they're typically in hypertonic environments. And if you expose them to fresh water or hypotonic, you don't get any more hypotonic than pure water. Water rushes into the cell. Now, a paramecium does have a contractile vacuole that can actively squeeze and pump water out of its cell. But if the rate of water moving in is too great, it's going to rupture that cell membrane. So we get conditions in different types of cells with cells having turgor pressure. If you're a plant cell, because it has full central vacuole, you can have situations in which cells shrivel, right? If it loses a lot of water in an animal cell, we call that crenation. So all of these water movements can have real consequences for really organisms as we go through. All right, time to practice. As I pull up the question, I'm gonna take a peek at the chat and see what I missed as we go through. So here we have starch and glucose with water. This is actually a really, really common lab that is done in some ways, trying to see how a fake cell, right, will change depending upon the certain conditions. So as you guys, thanks for cheering each other on in here. College Board, thanks for popping into the chat. Appreciate you checking in. Keeping up the good work, virtual teachers. Yeah, virtual, please give me a 5. I don't think it works like that, Valeria, but it's not, you know what? Maybe if you tweet at Trevor Packer and just say, hey, listen, I went to a live review at the Absolute Recap. I would love to get a 5. If the College Board would do that for me, that would be amazing. All right. Hello, AP Bio Penguins. We have a couple penguins in the chat. You did this lab, Angela. Yeah, I love this lab. There's a lot of variations of it in terms of describing the differences that you can see. So you might have done it with starch and glucose. You might have done it with salt. That's a really common thing for this as well. All right, so what do we think the answer is here? Which, excuse me, which is going to represent here the best justification for the movement that we see. And before you kind of jump into the answers, I see a couple of them happening already. Be mindful of your axes, right? What the axes are representing. Mrs. Jones getting a lot of love in the chat. Yeah, Angela, so your cells had food coloring. What happened in your lab with the food coloring? Were you tracking, was it an iodine lab? Were you tracking where the starch would be? Just curious. Looks like a lot of us have gotten an example here of what we think the answer should be. So let's look here at A and why we believe the answer to be A. Looking at our levels of water, starch, and glucose, we can see that starch has the relative amount in the bag staying constant, we see glucose decreasing and water increasing, and that's going to show the movement here. Now, starch isn't changing. Starch is huge. Starch is really large, right? Starch is a polysaccharide. It's not going to move through that dialysis tubing very, very easily, right? Maybe you poked a big hole through with a pencil or something, you would end up with some starch. Halftime break. We are halfway done. So I am 10 minutes farther than I thought I would be, but let's take a second. Where is everybody from? I'm kind of stealing. I think you can't see it anymore. I'm from Philadelphia. I have a Philly poster in the background of all my videos. But where are we located throughout the recap first? I don't think that's a thing. Throughout here, I got a message from someone who wasn't able to join us in the live today and was really hoping it would be recorded because it was 4 a.m. in Israel and they weren't going to be able to log in. Costa Rica, Houston, Austin, New Mexico. Awesome. Wisconsin. Love it. California. What time is it in California? And are you in the part of California where I'm going to be jealous of your weather? That'd be very sad for me. Although it was 80 degrees here today, so we'll take it. Georgia, 3:40 a.m. Martha, where is that? And why are you awake? I mean, thank you for coming. Oh, can't wait to see where Martha's from. But I really do appreciate you guys coming to this live. It's been a really, for me, a great experience to interact with all of you and hear about the questions that you have. One of the reasons you get into science is because students have an interest in finding out like what they don't know and how does all of this work. So it really is fantastic that you're able to dive into me. And if this is your last science class, I would be very sad. If this is your first science class, please take many more. But we are excited to have you here. Michigan. All right. Well, we do have India in here. Thank you. Oh, yes, Martha, make sure you sleep before your test. All right, guys, we're going to keep going. Take a quick stretch. Okay, quick stretch if you're sitting at the computer. Martha, you're in Africa. What part of Africa? There's lots of parts. Kyle, you're welcome. Monaco. I've been to Monaco. Monaco's fun. All right, if you haven't grabbed the study guide yet, it's not too late. Jump into the packet. So I have at the top of the chat that kind of pinned link for you. That's where you want to go. And the free preview, $0, right? Free preview includes more than just this study guide. So it's all of Unit 1 for free, everything that's in there. It's half of Unit 2 for free, and it's a couple other things in like Unit 3, Unit 5. So if you would like some extra things, you definitely want to grab that. Okay. All right, we are going to move on to everyone's favorite. I know that's not true. I'm pretending. All right, everyone's favorite, Unit 3, cellular energetics. I'm currently actually also teaching physics and laws of thermodynamics. Like, you just have to obey them. And Unit 3 is the emphasis and the example of how and why that's true. So cellular energetics is kind of a vast unit. Everyone typically just thinks of photosynthesis and cellular respiration. We will have a couple of concepts. Same thing, learning objectives. They're in the study guide. I'm going to skip them. Here's what you don't have to know. You don't have to calculate pH or Gibbs free energy. So if you took AP Chem and you are scarred, you don't have to do that here. But the next one's big. You don't have to know. Specific steps, molecules, enzymes of Calvin, Krebs, or any of the electron carriers or any intermediate molecules in the electron transport chain. That is huge because I do know that some teachers in AP Bio have asked that of you, and I don't fault them for it. I think I do a lot of it in my own explanation because I think it's a fuller picture of what's happening. But you don't have to. You're going to have information in the diagrams to reference should you need them, right? If they're going to ask you a question about the Krebs cycle, there's going to be a diagram most likely of the Krebs cycle for you to interact with in some way. The answer won't be overtly on it, but it should trigger your brain as we go through that. So I will come back to those when we get to those sections, but enzymes first. They're made of protein, so they have to follow all of the protein rules, we'll say. Active site for your substrate that is specific to the shape of that substrate. And if that active site changes shape because the protein has been modified either through an inhibitor that's allosteric or competitive, or perhaps the enzyme has been denatured in some way due to changing environmental conditions, that's going to be a problem. Now, oftentimes we use the expression that enzymes speed up the rate of reaction, and I'm using air quotes. The rate of reaction does typically increase, but enzymes just make reactions more likely to happen. And they do this in a lot of ways. But one of them that we typically talk about is lowering activation energy. And they make those reactions more favorable. They don't require as much energy to occur because they're bringing molecules together in a specific way. The two graphs that I have for us here do have Gibbs free energy on them, just available energy when we're working through a calculation. And if the products store more energy than the reactants did, that would be endergonic. And then we have exergonic when the energy is released to another thing, right? Not destroyed, but released to another thing. All reactions require energy to occur. It's just whether the products are going to be at a higher or lower energy state than the reactants. So enzymes in a nutshell. Personally, I don't really do this in Unit 3. I cover it with Unit 1 stuff with my students, but this is included here. AP Bio Penguin's asking if Winnie's going to visit. Winnie's currently snoring upstairs. I can hear her. That would be our bulldog mascot, but no, she's not coming. All right, time for a practice question. Make it big for you. Here we have the enzyme trypsin, which aids in protein digestion in the small intestine. No, you don't have to know anything about the small intestine to answer this question. But on my graph, I can see relative acidity and, relative activity and pH on my x-axis ranging here from 0 to 12. And I can see that the peak occurs around pH of 8. So enzymes are really picky. They're condition specific, right? So this particular enzyme seems to like a pH of 8. Now, why? Which of the following statements best explains the activity levels of trypsin shown in this diagram? That is a key part of this sentence. Shown in this diagram, right? So the information here has to link back to figure 1. So is it going to be because the small intestine releases inhibitor molecules? Is it going to be because there's just a number of effective collisions between trypsin and a substrate at higher pH? Is it going to be because as pH values increase, substrate concentration decreases? Or is it going to be that at low pH values, trypsin is denatured and cannot function effectively? All right. Energy cannot be created or destroyed. That's correct. Get a nice shout out. All right, we're all down there with D. Great job, guys. Looks good. I do want to pause here and just talk about collisions. So as you increase in temperature, I recognize that's not what this graph is saying, but as you increase in temperature, you do increase kinetic energy. And so you do increase enzyme activity because you have greater collisions between substrate and active site. But if you increase the temperature too much, you're going to end up with a denatured protein. So there's a fine line before we have to swap back to something different. There it is. Energy cannot be created or destroyed, just transformed from one into another. ATP. Answer the prompt. No, that's a different acronym, but I fully believe it, and I will talk about it on the final exam review day. This is our friend that carries energy from one place to another, our little energy shuttle. constantly removing that third phosphate to be a DP and an inorganic phosphate through hydrolysis, and then being reattached through ATP synthase in either of our electron transport chains. So we're going to see this guy a lot as our primary energy currency, as the analogy states. It's not, however, the only molecule that's going to transfer energy for us in these processes within our cells. Okay. Now, if you've been scarred by photosynthesis before, I'm sorry, but we can get through this together. As I stated previously, you don't have to memorize the steps. You also don't have to know many of the molecules. So the goal here is what goes in, what comes out, where, and what's the machinery involved in this process. And so the photosynthesis is broken down into lots of parts, meaning like it has 9 different names: light and dark reaction, light dependent and light independent, ETC and the Calvin cycle, tomato. It's all going to be describing the same function within different parts of the cell. So double membrane here, because the endosymbiosis theory, stacks of thylakoids, which have a lot of surface area, and then a liquid stroma. All right, starting on the left, then we'll go to the right, steps of photosynthesis. When we look at the overall equation, we want to find the players. Where are those pieces of the puzzle being used? Right, the goal of photosynthesis is to build sugar, glucose eventually. We are going to do this by inputting water and carbon dioxide and removing from this process, oxygen and our sugar. Don't fall in the trap. The gases don't go together, meaning water splits during the light reaction, releasing oxygen. When we have oxygen being released, we're not getting those oxygens from carbon dioxide. I know it can kind of be confusing in that way because we assume the gases go together, but that's not going to be how the process works. So light strikes the electrons, it excites them to a higher energy level, and they travel through the electron transport chain. Everything in green here is part of that chain, different shades showing different roles. And the roller coaster that those black dots, those electrons go on, is because they are losing energy as they pump hydrogens across the membrane, and they are recharged by in the first photosystem, which happens second, to transfer energy to our electron carrier, right, our NADPH molecule, which will shuttle energy over to the Calvin cycle. When we have this accumulation of hydrogen ions that build up, they are going to develop an electrochemical gradient. Great concentration, also electrically charged, and so they will flow passively back through the membrane through ATP synthase, and that's going to assist in the formation of ATP, that phosphorylation of this molecule. The Calvin cycle is going to receive energy from the electron chain and use that energy to physically manipulate these carbon molecules and form my 3-carbon sugar. So all of the carbons from carbon dioxide go into the Calvin cycle independently. Calvin cycle churns. We put together a G3P molecule, a 3-carbon molecule, in order to receive that product. So we actually have to do the Calvin cycle twice to end up with a 6-carbon sugar that will be formed at the end. So what goes in, what goes out, and why? The electron transport chain happens first to capture energy, transfer to the Calvin cycle. The Calvin cycle turns in the stroma, forming those sugars. Photosynthesis in a nutshell. All right, cellular respiration. 3 to 4 steps, depending upon how many breakdowns you want to do. The glycolysis step is going to be universal. All organisms can do it, whether you have oxygen or not, because it does not require oxygen to be accomplished. Even as it is the first step of cellular respiration, it is not going to have oxygen as a reactant yet. So when we look at the process of cellular respiration, oxygen does not actually have its debut until the very, very, very end of this entire chain. So if you have oxygen, we will take our broken down glucose molecule, our pyruvate, and we will, through a series of steps, oxidize it, and it'll go into the mitochondria. If there's no O2, we're going to follow a fermentation pathway. There's a couple versions here. We could do alcoholic fermentation, which will produce a carbon dioxide byproduct because we're going to remove a carbon in that. Or we might do lactic acid fermentation, which is a 3-carbon product. So we don't produce gas in that situation. But the goal is not to make ATP. There's actually no ATP made in the fermentation pathway. It's just going to regenerate NAD+. Right? So we formed NADH. We need to reform NAD+. And so fermentation pathways just allow glycolysis to occur again. Normally, NAD+, the enzyme carrier is going to, electron carrier is going to be recycled after it was used in the cellular respiration pathway. So we want to make sure that we send that shuttle back uncharged so glycolysis can happen again. So that's really the only reason fermentation pathways even produce any ATP. It's because glycolysis did it in the first place. Pyruvate oxidation it's a lovely phrase, right? It always gets overlooked when you go from pyruvate to acetyl-CoA. Yeah, glycolysis does make 2 pyruvates because if we look at the amount here, 6 carbons cut in half, 3 and 3. Each pyruvate molecule will go through the rest of cellular respiration independently. All right. So in photosynthesis, we went electron transport chain and ended with the cycle. In cellular respiration, we have the cycle first, ending with the electron transport chain. So the processes are like a mirror image. And this really reinforces the idea that we have reciprocal equations, right? If you flip it around, you have really, really concrete pieces. So what goes in, what comes out, where, and why? When we look at the Krebs cycle, we are in the mitochondrial matrix, right? The fluid portion of the matrix. So we have cycles happening in the fluid portion of each respective organelle. And the goal of this is to physically continue to break down all of my carbons from my original glucose molecule. So pyruvate gets oxidized to acetyl-CoA. Acetyl-CoA is going to go into the cycle, and we're going to remove the carbons as this cycle churns, which is why carbon dioxide leaves the Krebs cycle, right? CO2 comes off. We also are going to get some electron carriers that will help us move through the electron transport chain, which is going to occur in the inner mitochondrial membrane. But the process is really similar. We drop off electrons from the electron carriers. They go through the chain. The movement of those electrons actively pumps across hydrogen, similar to how we saw in photosynthesis. We build up an electrochemical gradient, and they flow back through ATP synthase. So all of those kind of big picture concepts still play out in cellular respiration. As ATP synthase has the hydrogen ions moving through, we are going to assist in the formation of ATP. All right, let's practice. We are almost done. Unit 3, hitting the hour mark, so we want to wrap up shortly. It's estimated that oxygen production first evolved in photosynthetic prokaryotes approximately 2.7 billion years ago. The first photosynthetic prokaryotes are presumed to be similar to today's cyanobacteria. Which of the following best supports the claim that the prokaryotes were responsible for the oxygen in Earth's atmosphere? All right, what do we think? So if you think you know, pop it into the chat. Yes, I see some things in the chat talking about NADH and NAD+. Reduction is when it does gain that electron and hydrogen. Yeah, so we lose the positive in that. So we think of the NADH like holding on to the energy, and NAD plus is not. NADH in cellular respiration serves a nearly identical function to NADPH in photosynthesis, and they both are going to be electron carriers for that. Looks like we are split a little bit between A and B. Let's take a peek. B is going to be, excuse me, our correct answer here. As we look ahead, really the question is asking like, where did the oxygen come from? And so if we look at the process and look at the steps, that's going to be where we see our oxygen from the splitting of water when it first strikes the photosystem. Okay. Last unit, and this is going to be a short one as we move through. So unit 4, cell communication and cell cycle. One big game of telephone. Should be simple, right? All right, learning objectives, again, they're on your study guide. Grab it in the chat if you haven't. Here's what you don't need to know. You don't need to know any specific cyclin CDK pairs or growth factors. So even within the cell cycle, excuse me, in the cell cycle and within diagrams, they're going to be labeled for you, but you don't have to know them specifically. Thank God for the mute button, I'll tell you that much. All right, 3 primary steps: reception, transduction, and response. I grabbed this diagram on purpose. If you look down in the corner, you can see where it came from. 2022 AP Bio FRQ number 1. One of the challenging things about signal transduction pathways is how you're not required to know any specific pathway. It would be so much easier if you could just like have one in your brain and just use that as your example every time. That's just not the way that it works. And so this particular diagram I really like because, they're going to show you a couple of different options of how things become activated and inactivated, how they relay information from one area to another. All of which starts with a ligand. So the ligand will bind either extracellularly or intracellularly based upon its own chemical makeup. So in this diagram, we have a extracellular ligand. That would imply that it's not nonpolar, or maybe it's really large and it can't fit through the membrane. When we have transduction, we can be involved in taking a really small signal and just causing a significantly large relay system. This might involve several protein kinases. It might involve phosphorylation cascades. It might involve secondary messengers. So a secondary messenger would be something that perhaps calcium, maybe the ligand itself didn't cross the membrane, but it caused a channel protein to open. And then you had literally a secondary messenger crossing the membrane to then continue the signal. So there's a lot of variation here. My best advice when it comes to cell communication pathways is just to look at a lot of examples. And the best ones are, in fact, in the CED, looking at the ones that are in FRQs that have already been released, and then the response. Really common to have responses that are protein synthesis related. Sometimes the responses will be vesicle formation related. But in many ways, cells have to communicate about lots of things. So on the exam, they're just going to have to give you enough information for that question to be answered. Just reference back the diagram and use the terms off the diagram itself. but let's give it a go. So insulin is a protein hormone that is secreted in response to elevated blood glucose levels. When insulin binds to its receptors on liver cells, the activated receptors stimulate phosphorylation cascades that cause the translocation of glucose transporters in the plasma membrane. All right, what's the role of insulin in this paragraph? What is the role of insulin in this paragraph? If you think you know the answer, pop it into the chat in this example. This is a good representation, though, of the fact that all of these questions will have real-world application. So they're not going to be made up. They will have real-world applications. All right, overwhelming response of A in the chat. Great job. Protein hormone. So it's going to bond. It's going to bond extracellularly. It's not going to be able to diffuse. Feedback mechanisms. 2 types, negative and positive. Key thing here is that it's going to have some information relative to homeostasis. So homeostasis is a buzzword for you, but you're kind of blanking on the definition. Let me just regroup it. So homeostasis, the maintenance of constant internal conditions despite external changes. So this is the body's ability to respond to stimuli from its external environment as well as internal conditions. And all of that is to kind of keep it in its Goldilocks zone. Most feedback loops are negative. It's going to bring the organism back to homeostasis, back to the area where it is most functional. Positive feedback loops are very temporary, but they have a really strong purpose in allowing organisms to typically thrive in stressful or like life or death or reproductive temporary moments. And so there's not as many examples of positive feedback loops. Childbirth, lactation in mammals is a good example as well. And the way that your blood clots is also really a good example. Let's do a quick practice. All right, here we have some blood clotting. Which of the following best explains the feedback mechanism based upon the picture? I see things labeled like smooth muscle, collagen, endothelial cells. Endothelial cells is probably something you didn't study. You likely also may not know blood vessel structure. But what we do see here is chemicals being released. We see a circle, an arrow kind of curving back around, showing that continuation as we go. So which explains the feedback mechanism illustrated in this figure? Okay, I see a couple in the chat. Some C, some A. All right, so it is A, but let's look at C. So this is an example of negative feedback because a large clump of platelets can block the blood vessel and prevent blood flow through it. So the curved arrow here is an intentional process. We have to remember that the intentional process of returning to set point, returning to normal, wouldn't really be describing negative feedback, because negative feedback, if it impedes the blood flow, that's not going to be a homeostasis condition. So that would be why C is not correct. Okay. The reason why it's A is because platelets are causing more platelets, and this clotting is not a standard condition of the body, right? And so we have a leak in the blood vessel here. These chemicals are causing more platelets to come and close that hole. We wouldn't typically want that to occur within an organism. All right, cell cycle checkpoints. Looking through here, we have interphase consisting of G1, S, and G2. There are a lot of checkpoints within here to ensure the cells should continue throughout its growth process. The way that this is described, and you often see M phase identified with mitosis and then cytokinesis, they are separate processes, but they have to ensure that the right parts are connected. So often we're going to look for damage. Do we have spindle fibers attached? Do we have all of our chromosomes? Have they been replicated? Are we ready for the next phase? Not shown here is G0, but G0 is that non-dividing stage that some cells will go into instead of continuing through the S phase. All right, and lastly here, mitosis. Mitosis, looking through and distinguishing between different parts and pieces. So remember, mitosis is a process of cell division as we move through PMAT, prophase, metaphase, anaphase, and telophase, to divide chromosomes evenly and create diploid cells. So the sister chromatid structure, the X that we see in mitosis, is going to pull apart an anaphase, and it's going to allow identical sister chromatids that were formed in the S phase to migrate to opposite poles of the cell. This is possible because during metaphase, all of my sister chromatids line up in the middle, spindle fibers shorten, pulling those components apart. So when we get to meiosis, which we won't until unit 5, we'll notice that the chromosomes align in the metaphase differently. And the way that my pairs separate will be by homologous. Here I'm separating sister chromatids. In mitosis, I'm maintaining my chromosome number. So diploid 2N, I'm still diploid 2N in the resultant cell. So I need to form identical cells at the end of mitosis. So to recap, we have neared the end. 4 units here, looking through CHNOPS, that we have proteins, carbs, lipids, nucleic acids. Water is amazing and it's polar. In cell structure and function, we have surface area to volume ratio, we have organelles that are involved, and cells really compartmentalize their functions to transport materials in and out. Cellular energetics, don't break the rules of thermodynamics. Energy cannot be created or destroyed, but it's transferred. Sunlight into sugars is photosynthesis, sugars into ATP is cellular respiration, and enzymes are reusable. They're not going to be destroyed in a chemical reaction. They're not a reactant. They are substrate and condition specific. Lastly, in cellular respiration in the cell cycle, we have the reception, transduction, and response. You have feedback loops with homeostasis, and cell division and checkpoints with mitosis and PMAT. So coming up next on the absolute recap, we have the May 8th, which is tomorrow night, practice test. Next week, we will, yes, I have a question in the chat. We will do units 5 through 8. And then on May 15th, we will have a final review. There'll be 2 of them. The one on YouTube will start first, and then there will be a live stream for Ultimate Review Packet subscribers only. That will be a little bit longer and occurring after that. So I want to say thank you so much for coming, and I hope that you found this helpful. I did not quite stick to the hour mark, but I appreciate all of you coming to study with me and your participation in the chat. Your support of each other is absolutely amazing. I really feel strongly about this community that we have here at the recap. And if you think this was helpful, right? If you think that you would like to see more of this in the future, whether it be for bio or a different subject, if you think that you want to invite a buddy to the next one, happy to have you. And we really can't wait for, well, I can't believe the exam is coming up next week. This went so fast, so fast. It is Teacher Appreciation Week. So if you want to say a quick shout out to your teacher in the chat, I'm sure they would absolutely appreciate it. They are working really, really hard in these last few days. They're equally invested in your success. And I know that they want to see good things for you. So Again, thank you so much for coming. Yes, and those of you who are up at 3 in the morning, absolutely impressive. I think all of you are going to do good things moving forward. So, with that, I will say, have a great night.